A-level biology students, could you explain how negative feedback is involved in control of blood glucose concentration? Here's what you need to know. The pancreas contains alpha and beta cells which act as receptors. They detect changes in blood glucose concentration and respond by releasing hormones. Let's start with high blood glucose concentration. When blood glucose rises above the optimum concentration, for example after a meal, beta cells detect this change and secrete the hormone insulin in response. Insulin is transported in the blood and binds to specific receptors on the cell surface membranes of its target cells, which are skeletal muscle cells and liver cells. When insulin binds to insulin receptors, the cells insert more channel proteins into their cell surface membranes. This increases the uptake of glucose into the cells by facilitated diffusion, lowering the concentration in the blood. Inside the cells, insulin also activates enzymes involved in glycogenesis, the synthesis of glycogen from glucose. So all that extra glucose gets stored away. Now let's look at low blood glucose concentration. When blood glucose falls, for example during exercise, alpha cells in the pancreas detect this change and secrete the hormone glucagon in response. Glucagon binds to specific receptors on the liver cells, which are its target cells. It activates enzymes involved in glycogenolysis, the hydrolysis of glycogen to glucose. It also activates enzymes involved in gluconeogenesis. This is the conversion of glycerol, fatty acids or amino acids into glucose. This glucose can leave the cells and enter the blood, raising the blood glucose concentration. So to summarise, insulin decreases blood glucose by increasing glucose uptake into cells and promoting glycogenesis. Glucagon increases blood glucose by promoting glycogenolysis and gluconeogenesis. Check out the caption for breakdowns of these long G words.